The overall goal of this procedure is to dissect and mount zebrafish embryos for optimal microscopic visualization of the spinal cord. This is accomplished by first microdissecting and removing the yolk ball. The second step is to remove the yolk debris and pipette the embryos onto a slide. Next, the excess liquid is carefully removed with a Kim wipe, followed by addition of mounting medium. The final step is to orient the embryos on the slide with an embryo poker, followed by application of the cover slip. Ultimately, fluorescence or bright field microscopy is used to show distribution of postmitotic cell bodies and the trajectory of developing axons.
This method can help answer key questions in the field of neurobiology, such as what are the mechanisms responsible for patterning, differentiation, and axon guidance.
Though this method can provide insight into many aspects of nervous system development, it can also be applied to other tissues, such as developing muscle.
Generally, individuals new to this method will struggle because dissection under a microscope is required.
Prior to beginning the experiment, make an embryo poker by gluing a pipette tip to a Pasteur pipette and then gluing a fishing line to the pipette tip. To begin the embryo dissection, place up to 30 zebrafish embryos aged 24 hours post-fertilization into a 35 mm Petri dish filled with PBS. Under a dissecting microscope, secure an embryo's head with a pair of forceps while pulling the yolk away with an insect pin. Then use the embryo poker to move the embryos to a part of the Petri dish that does not have a lot of yolk debris.